Good afternoon, colleagues. Today we have an opportunity to listen Mywish Maredia. She is a professor of international development at this Michigan State University, and she holds a PhD from the same university in 1994, and she obtained her bachelor's from this University of Bombay. And she also got from her PhD thesis the American Agriculture Economics Association this outstanding thesis award. But that is a long history, which a long back to have that one. She has worked a lot on this impact assessment and development of innovative tools and methods for adoption, this impact assessment, and throughout those Asia, Africa, and Latin America, she has been able to implement it on different ways, and many publications has come out from those one. I am one of the beneficiary from her this research efforts which she has done. She does not know, because today only the first time I have met her face to face, and in between never I have interacted with her. But she did a wonderful study which was published along with this Derek Barley in this agricultural economics, that was quantifying the spillover impacts, econometric estimation on this wheat research conducted by to have that one. I have implemented that one along with other one, and quantified the spillover impacts of this sorghum research and ICRISAT, and it gave me also a good way to the fame. This standing panel of impact assessment, this PIA of CGIAR, they have evaluated for this 2003 to 2006 all the CG centers which they have done those impact assessment studies, and they have found these 6 studies to be exemplary one. One was that was having that one, which was by me along with Dr. Cynthia Vantilan and others. So I take this opportunity to have that one, that intellectual debt which I had to you. And the one which why I wanted to mention that one, that at that point in time, that econometric estimation method which she was able to publish was an innovative way, and which others could not think of that one. Today what you are going to listen that one, you will be finding that 10 years from now, it would be having this forefront in terms of this adoption and this impact assessment one, new protocol, new tools which are having that one. So I am telling that one, today's this, you would be part of this history of this development of new methods and tools, which she is going to tell us, and we would take this opportunity to listen. And we have not only the people who are in front of us, also those people who are having in the global community, which are connected through this internet, KSIConnect, and they are also listening that one. With this one, I would request Mywish to present her this important seminar.
Okay. Thank you very much, Uttam, for that introduction. I'll go right ahead into the presentation, first by acknowledging all the collaborators. This study that I'm going to present today could not be done without collaborations from a multidisciplinary team, and I have just listed all the people that have been part of this project of the 2 case studies I'm going to present. And Byron was one of my colleagues at MSU, now he's at CIAT. For Cassava case study, we worked very closely with Crop Research Institute in Ghana. Joe Manu is the Cassava breeder. Avere Danchi is the socioeconomist. Then from IITA, our collaborators were Peter Kulako, Ismail Rabi, Elizabeth Parkis, Tahiru Abdullahi, and Gazahian Girma, and from Cornell Ramupunna. For the bean study, we have worked very closely with the Zambia Agricultural Research Institute, and Petan and Kennedy were our 2 collaborators. Kennedy is the bean breeder there, and Petan is the socioeconomist, and also with CIAT, and Enid, Bodo, Claire, and Alan. I just listed them as sort of the main contributors to this study. Also wanted to acknowledge upfront, the funding came from the Standing Panel on Impact Assessment of the CGIAR through the SIAC grant, which is SIAC stands for Strengthening Impact Assessment in CGIAR. And I fully acknowledge that this is part of that SIAC program, and the funding was, it was part of our work plan under that program. Also, partial funding came from the CRP on roots, tubers, and bananas, and also we heavily relied on the technical support from IIT and SIAT. Of course, also thanks to all the enumerators and field staff and farmers, respondents who participated in our study, and for sharing their time and knowledge. Although, I heavily acknowledge and depend on the technical expertise and knowledge contribution of all my collaborators, I just wanted to make a big disclaimer that whatever results I'm going to present, this is the first time I'm presenting the results. Any errors of omission, commission, or interpretation rest with me. The outline of my presentation will be, I'll just give a overview or rationale for why we are doing this study, the methodology involved, present some results, and end with some discussion points. So in terms of the rationale, I guess we all are in the business of doing research, and our research is funded by public sector, the donors, and they keep on asking us, show us the impact, show us the outputs and outcomes of your research. So I don't think I need to say much to this audience in terms of the importance of why we need to be measuring varietal technology adoption. Just to leave it there, that it's very important as a CGC for the CGIAR as well as for national programs who are funded by governments, public sector agencies, that we need to be accountable and show what our results are, especially for plant breeding research, which has been one of the flagship program of CG system. And we have claimed to have, released so many varieties either directly or indirectly. And over times, donors have been asking us to continuously track the adoption of these technologies at the farmer level, not just how many were released by the national program, but how many have been adopted and are being used by the farmers. So I don't think I have to spend more time in terms of justifying the importance of technology adoption. So in terms of what methodologies have been used in the past or currently are being used to measure the varietal adoption, there are many methods that we socioeconomists routinely use, and this include using secondary sources. Many times government agencies routinely collect data and publish them in reports in terms of the adoption of different technologies, and we rely and use those sources. For varietal adoption, we also many times have used seed sales data or data on the actual seed distribution to estimate what is the adoption of different varieties. Another method that have been used is expert elicitation, basically bringing together experts who are knowledgeable about the adoption of varieties in specific geographic areas, bringing them together, doing some brainstorming session, and coming up with a consensus of, okay, what are the adoption rates of different varieties in specific geographies, and then sort of aggregating that to either a state level or a national level, depending on the size of the geography. There have been also community level surveys that have been used by us, the socioeconomists, whereby we have focus group discussions at a village level and elicit this information from the village leaders as to their perception and knowledge of what the adoption of different varieties of a specific crop is in that village. And then lastly, farmer elicitation method, which is basically as part of the household or farmer survey, we include questions on asking directly to the farmers what varieties they have planted or have been planting, depending on the time frame that the study would be focusing on, and recording the names of the varieties as reported by the farmers, or as at least the type of varieties, whether it's a modern variety, traditional variety, and then using that information to estimate adoption level for specific crop and varieties. Each of this method has pros and cons, so there are challenges in using in each of these method, and usually the challenge is the trade-off between the cost of collecting such information versus the accuracy with which you can collect. So if you look at the farmer level survey, we can think of that as giving us the most accurate information because you're going to the adopter himself or herself and asking that question. But doing such survey at a representative scale is a very costly affair. So there is this trade-off. Whereas on the other hand, doing expert elicitation, maybe less costly, but the type of information you get may have a higher confidence interval. You're not very sure whether the estimates you're getting through that method, how close it is to the actual reality on the ground. Okay, so there is usually this trade-off, and also the trade-off between cost and frequency. If the method is a high-cost method, you won't be doing it as frequently as you want to, or there is demand for doing this at least periodically. Every 3 or 4 years you want to update your estimates of adoption of varieties. But if the method is very costly, you may not do it at that frequently. And there is also difference in these methods in terms of their credibility. Some are considered more credible and rigorous than others, and thus not frequently conducted at a representative scale. The farmer elicitation method, which is going to the household, as part of the household survey, interviewing the farmers and asking the questions, is currently sort of considered the gold standard or the most rigorous way of collecting, estimating the varietal adoption. But the question is it really a gold standard? Okay, is all that glitters gold? Is going to the farmers and eliciting this information actually give you an accurate picture of varietal adoption? And I just wanted to point out few confounding factors that can limit or can make this a questionable methodology. There are first of all, many practical challenges about using this method. That when you ask a farmer what variety he or she has planted, the response you get from them may not give you a true picture of actually what variety they are using. First of all, they may give you local names, and those local names cannot be matched with your official varietal release list that you have, and so you still don't know actually what variety that farmer had planted. Or farmers may not able to identify the variety by a name, or may just say, I don't know. Many times in our surveys we just have responses which they say don't know. And when we get such responses, again, they don't provide useful information to estimate adoption rates. Many times they may give you a name, but it can be, not the correct name, even though they may claim that to be an improved variety. And also many times, even if you just ask whether you have grown an improved variety or a local variety, many times they don't know what is an improved variety, and so you cannot rely on their response. And for many crops, such as maize or OPVs, cross-pollinated crops, even asking farmers what variety they have planted, and they may even give you an accurate response, but if they have recycled the seeds for several seasons, that variety may not be that variety anymore. From a genetical perspective, they may have lost their identity. So all these are sort of the problems or challenges of relying or using the farmer elicitation method, which has traditionally been used by us economists. And so under the SIAC project, we wanted to sort of test the accuracy of this method plus some other method in comparison to the method that has been used, so that we can sort of estimate the direction of the bias and the error that are part of the methods that have traditionally been used. So that error and bias remains unknown in the literature. People have used these methods, but nobody knows how accurate or close to the accurate estimate they are. And thus, we wanted to test these different methods and approaches in terms of correctly identifying a variety. And to do that, DNA fingerprinting sort of offers that reliable method against which we can test the accuracy of these different methods. And the use of this DNA fingerprinting can, if we use it in conjunction with these other methods, can increase the accuracy and credibility of the data we get, and also the interpretation of that data for economic analysis. And also DNA fingerprinting can serve as a benchmark against which we can validate other methods which may be less costly, and so we can find a more cost-effective way of estimating varietal adoption. And this particular aspect is important when we think of scaling up this to a larger geographic scale. So we all know DNA fingerprinting's the importance of it and what it can offer. But however, despite these advantages, this method is not widely used as of now for tracking varietal adoption by farmers. There are questions related to the sampling and logistics, and also the cost effectiveness of this method, which has not been explored rigorously by anybody so far. So under the SciAC project, 2 pilot studies were conducted, one in Ghana and another in Zambia, to sort of explore and understand the practical application of this DNA fingerprinting method for varietal identification, and also to test the effectiveness of alternate methods of varietal identification against this benchmark of DNA fingerprinting. And third, to come up with some lessons learned and recommendations on methods and approaches for scaling up. So the 2 crop country combinations for this pilot study, in Ghana we focused on cassava, and in Zambia on common beans. And these 2 crop country combinations were selected basically based on whatever opportunities were out there for us to sort of piggyback on those plant studies and add this component to it. So this was specifically the case for the beans in Zambia, where adoption survey was already planned, and we just piggybacked on that opportunity and added this fingerprinting component. The second reason for selecting cassava versus beans sort of was to also make sure we have diverse crops. Cassava is a root crop, beans is a field crop, and also wanted to see how this methodology works on this kind of a root crop versus sort of a field crop. And also there was interest from national and national centers and CGIER centers to collaborate on these 2 studies, especially because it requires a multidisciplinary team of researchers, and we had such demand and interest from institutions that do research on these 2 crops. So in terms of methodologies, as I said, one of the objectives of this pilot study was to test different methods of collecting adoption data. So in this table we sort of summarize the methods that were used for both cassava and Zambia and beans in Zambia. And this include in both the countries we used, of course, DNA fingerprinting as the benchmark method, and also in both the countries the current gold standard of farmer elicitation method was used, whereby farmers were just asked what name of the variety they were planting, and or the type of variety their knowledge about whether it was an improved variety or traditional variety. In the case of Ghana, the Cassava case study, we also included in our household surveys a method which has never been used before, which was to show them a series of photographs which showed different morphological characteristics of the plant, the cassava plant, at different stages, growth stages, and then asking them to to pick the characteristics that best matches the varietal characteristics of what they were growing. Yeah, so we showed them pictures of, let's say, when the plant is very young, whether the leaf, whether the plant had pubescence, what characteristics were there, then when it was 3 to 6 months, what was the color of the leaves, the shape of the leaves, the way it was branching, the height of the tree when it's mature, and things like that. So some of, there were about 11 or 12 characteristics. The pictures were shown to the farmers, and depending on the options they selected, the experts of cassava tell us that those combination of different morphological characteristics can help tell whether that variety the farmer had used was improved variety or traditional variety. Okay, so that was the method we used in cassava in the case of Ghana. The method C was very similar to method B that we used for cassava, but in the case of beans, the enumerators carried with themselves samples of seeds of improved varieties in small plastic bags, and at the time of the interview they showed those samples to the farmers and said, okay, which of these samples matches the varieties or the seeds you had planted? Okay, and then the farmer would say, okay, that one, and they were all coded, the names were not there, and then those codes were then translated into varieties. Okay. In method D, this was only used in the case of cassava, a sort of a cassava expert or somebody who knew the cassava crop very well, accompanied the team of enumerators or the team of his survey team as part of the survey team, and they visited the field in which the cassava was planted, and they observed the same morphological characteristics, recorded them, and later that information was to be used to see what combinations of these morphological characteristics match with the library of characteristics of the released varieties, and see if they could be identified using those combinations. While they were in the field, based on their observations, that expert also recorded his opinion of what that variety was. Yeah. So that within this method there were sort of two methods. One was recording the morphological characteristics, and second, the cassava expert himself or herself giving the opinion of what that variety was. The method E was used in both the countries or for both the crops. Here, during the interview or the household survey, the enumerators in the case of cassava, when they were visiting the field, they took pictures of the cassava plant specific to that variety, and then those pictures later on, after the survey was done, was shown to a panel of experts, and based on the pictures, the experts gave their opinion of what that variety was. In the case of beans, the enumerators took pictures of the seed samples that the farmers showed, and then later on, after the survey, the pictures of those seed samples were shown to a panel of experts, and based on those photographs, the experts gave an opinion of what that variety was. In the case of beans, the enumerators also collected the samples, the seed samples from the farmers, and that seed sample was collected also for DNA fingerprinting, but it also served a dual purpose of using in the method F, which is that those same experts that looked at the pictures and gave their opinion on the variety, the next day they were also shown the seeds So they actually observed the seed samples from the farmers and then gave their opinion on what that variety was. Okay. So these were some of the methods we tested in the two crop country combinations. And a footnote regarding method A is that in Zambia, the enumerators or our team also collected some samples from the market. So they went to some bean vendors and asked them to share some samples of the beans, asked them what variety they think the sample, the seed was, and then those seeds were also included in our study. So let me give you some details on what was our sampling and data collection method for these two case studies. In the case of Ghana, the study was conducted in 3 regions, and these regions were selected based on the importance of cassava. Together they cover about 61% of cassava production, and these were the Brong-Ahafo, Ashanti and the eastern region of Ghana. A total of 500 households across 100 villages, which means 5 farmers per village, was targeted for the survey, and these farmers were selected using a multi-stage cluster sampling method where the district, village and farmers were randomly selected. The survey took place in October to November in 2013, and it was coordinated by a research team led by the Ghana Crops Research Institute. So in the case of Ghana, the enumerator team, the survey team included 3 people. One was the enumerator who was in charge of doing the household module, the interview with the farmer, a cassava expert who was in charge of completing that field survey module where he or she went to the field and recorded the observations, and also there were some field related questions. And there was also a DNA sampling expert included as part of this team, and that person was in charge of collecting the samples for DNA fingerprinting, labeling, making sure they are properly labeled and stored as per the protocol that was established. In the case of Zambia, the study took place in 2 northern provinces, Muchinga and another province also called Northern Province, and again these were selected based on the importance of beans and also the prior seed dissemination efforts that had taken place. The study, as I told you, was designed to take advantage of an already planned adoption survey by PABRA, and it was being conducted by ZARI. There were totally 7 districts were purposely selected, and they together represent about 60% of total bean area in Zambia. In this case, our sample in size was 400 farmers spread across 67 villages, and this sample size was mostly determined based on the available budget. The survey was implemented in August-September of 2013. In the case of Zambia, the enumerators were trained, so there was only one enumerator that did the survey, which is very traditional how socioeconomic surveys are done. So this was very similar to the current method of doing farmer elicitation. The enumerators were trained on how to use that instrument, take photographs of the seed samples, collect the seed samples for both DNA fingerprinting as well as for one of the methods of varietal identification. And also each enumerator carried with carried with him or her the seed samples of about 10 released varieties in Zambia, and this was to implement the method C. In terms of the methods E and F, which are, just to recall, these are the expert elicitation method, whereby the experts looked at the photographs or the seed samples and gave their opinion on what that variety was. This was conducted ex post of the survey. So once the survey was completed, and several months after that, A panel of crop experts were brought together, and this, in the case of cassava, the experts included the breeders and the extension staff from those study districts. I'm sorry, in the case of beans, and then in the case of cassava, the experts included the cassava breeder and experts and field technicians from that research institution, also from IITA and a university of Cape Coast. In both the cases, the overall elicitation process was facilitated by a socioeconomist whose main role was to just facilitate that process, but not to give his or her own opinion. Okay, so he did not take part in the identification, just served as a facilitator for that method. And the way the expert elicitation outcome was recorded was little different in both the cases. In the case of Ghana, the cassava experts recorded individually their opinion on what that variety was. They never came together to reach a consensus. Whereas in the case of Zambia, the panel of experts sort of had discussions, debate, and then they came together as a consensus what that variety was, and noted both the name of the variety according to their experience and expertise, and also whether it was an improved variety or a local variety. Okay. In the case of Ghana, the experts only recorded whether it was an improved variety or local variety. They did not indulge themselves in giving the name of the variety. So in a way, there was sort of nuanced differences in the way this method was implemented in the two countries. Coming to the DNA fingerprinting method, as I said, in both the countries, this method was used as the benchmark method to validate and evaluate the other methods. And this method first involved establishing a reference library of DNA fingerprints, and then applying the same or a subset of markers that were used to establish the reference library to then genotype the samples that were collected from the farmers through the farmer surveys. So in terms of the sampling method for DNA fingerprinting, we had little different methods in both the countries, and this was mostly dictated by the nature of the crop. So in the case of cassava, which is a root crop, so there are no seeds, we collected the leaf tissues, and as I mentioned before, the team included what we call a cassava expert, who was basically field staff of the crop research institution, part of the cassava breeding program there. And this expert visited one of the fields of the farmer where most varieties of cassava were grown. So in terms of our sampling, at the household level, we asked the farmer how many fields they had planted the cassava. If the farmer had planted cassava in multiple fields, then we asked which field had the most varieties of cassava, and we visited that field and sampled the varieties from that field. Okay, so just keep in mind that even though we have 400 or so, 500 so farmers in our sample, and they may be growing cassava in multiple crops, we only ended up visiting one field per farmer, per household. However, in that field, all the cassava varieties that he claimed he was planting, the farmer was planting, we collected the samples. And the way we collected this, the method we used was the cassava expert took one. So if this is the field of cassava, and the farmer said, half of this is variety A and another is variety B, then for this part of the field where variety A was planted, the expert randomly selected one plant and picked up the youngest leaves, the apical leaves of that plant, and put it in a bottle where there was silica gel, and then it was sealed, and that bottle was labeled with the household ID, the variety name, and the variety ID. Okay, and the same method then was used for the variety B, if the farmer had planted 2 varieties. However, the reason we wanted to include a cassava expert is that we were told by cassava breeders that the farmer may say he or she is planting 1 variety, but in actuality there could be natural variation of that variety in the field, and they wanted to capture that the genetic diversity within the field if there was some natural variation. So the cassava expert, when he went to the field and observed some natural variation of the variety, even though the farmer said it was 1 variety, but according to his opinion, the expert's opinion, if there was some natural variation, they also sampled the leaves of that plant, which appear a little different in terms of morphological characteristics. Okay. So this was sort of the method we had used, the silica gel in a plastic jar, and within a day or 2, the leaves that were collected had already dried and were ready for, this increased the shelf life of the samples to do a DNA fingerprinting. So in terms of the field logistics, as you can imagine, a lot of things need to be in place, need to take, in terms of making sure you get the viable samples for DNA analysis. It's very important because, the collection of the samples from the farmers' fields to making it available or sending it, transporting it to the labs, there could be hundreds of miles of distance between that, and we needed to make sure that there was a proper protocol set. All the enumerators and everybody involved in that chain, the value chain, were well trained on their role and what they needed to do when the samples were handed over to them from one step to another. Okay. There was also a versatile and economical sampling kit we had to develop to make sure the samples were collected. The process was not too complicated because this is all done at the village level or at the farm field level. The samples were then shipped to IITA where the DNA was extracted, and they did this more than 1,000 samples in less than 1 week. All the DNA was extracted from the farmer samples. The DNA was then freeze dried and shipped to Cornell for the genotype by sequencing analysis. So to establish the library, a total of 64 accessions of released varieties, 18 released varieties and 46 land races were included in these reference libraries. So to recall, as I said, we have to first establish a reference point against which the farmer samples would then be compared. So this is what we are calling the reference library, and in the case of cassava it included the 64 accessions. Then these samples of the 64 accessions of the reference library along with the farmer collected samples were genotyped at 56,849 SNP loci. Don't ask me much about what this is. I'm sure there are experts in this room who can explain more what this is. I'm just repeating what I've heard. Then in terms of the analysis, or actually what was done was using the GBS technique, the genetically identical sets of clones were identified using this distance-based hierarchical clustering model-based maximum likelihood admixture analysis method, and this was done by researchers at IITA. In the case of beans in Zambia, our sampling and logistics was a little different, again because it's the nature of the crop. Here seeds were available, the time when the survey was done, the farmers had already harvested the crops, so there was no option of doing the leaf tissue collection. So we collected the grain samples in this case, and the seeds that were collected from the farmers were later on then germinated in the greenhouse by the bean breeder, and leaves were collected from those germinated young bean plants and put into a leaf collection kit that we got from the LGC Genomics laboratory, and then those plates which had 96 wells with the samples were then shipped to them, and then they did the analysis. Additionally, 20% of the bean samples were again germinated at Michigan State University. We just wanted to replicate some of these samples just to make sure we can have good confidence in the results we are going to get from that original sample. So in the case of beans, regarding the reference library, we included 13 accessions which were released varieties in Zambia, and 2 land races of a specific or a very popular market class called Kablungeti. So this was our Zambia specific reference library that we included. In addition to this, we also used about 723 accessions that were collected as part of a another SIAT project within the East and Southern Africa region, as sort of the background material or serve as a sort of a very broad reference library to compare our Zambia collected samples against that. Okay, so the farmer samples were genotyped using 66 assays or markers, which were selected as a subset of the 800 SNPs which were used for the reference library, and these 66 SNPs assays were selected or made up of 4 groups, each of which had more or less the same power to differentiate the released varieties from each other, because it was very important to differentiate each of those reference library materials, and also the background genotypes that were included. Again, in this step, the SIAT researchers were our main collaborators and helped us do this analysis of the genotyping data. So some of the sample characteristics of our 2 country case studies. In the case of Zambia, we had 402 farmers that we had surveyed. On an average, about each farmer planted beans on 1.36 plots, and on an average they planted 2 varieties per household. The average number of the number of varietal data points that are part of our analysis are about 863 in the case of beans. They are about 924 in the case of cassava, and the number of samples that were genotyped for the benchmark method of DNA fingerprinting, they are similar but not the same. And the reason is that in the case of beans especially, even though farmers may have planted 2 varieties, by the time we went, some of them had already sold their harvest, so they didn't have samples to give it to us. So that is one of the limitation of using the seed-based sampling method, where you do risk not collecting the sample, because if the timing is such that the farmer has already sold the harvest, then you don't have anything to sample. So that was sort of one of the thing we, sort of a lesson learned kind of thing for us in the case of beans. In the case of cassava, there were also some, lost of samples in this whole process. So we have 917 samples that were genotyped at the end. So results. Just asking the farmers what type of variety you planted. So in terms of results, I'll first go through all the results related to beans and then to cassava. So for beans, when we asked them what type of variety you had planted, out of 802 data points of varieties we have, 106 can be classified are reported as improved varieties, 612 as local varieties, and 84 don't know. Okay, so they didn't know which type of variety they were planting. In terms of variety specific responses, across the 4 methods we had used, asking the farmers, the name of the variety they had planted. You can see a very small number of responses match to the variety release list that we have for Zambia. Okay, so for example, out of 855 data points we have in our sample, only 1 matched Lukupa, only 1 matched Lambai, only 1 matched Kapisha and Lwangeni. These are the names of the released varieties, and about 30 matched Kablumgeti. Okay, so you can imagine, I mean, this is a very low. If you were just to use this method, you would end up concluding that the adoption rate of improved varieties is, dismal, very, very low. Okay, in the method C, when we showed the samples to the farmers and asked them, tell us which variety that you planted matches the seeds of this bean seeds, we got very diverse and more variable response, and it's more than a majority of farmers. There is sort of variety of responses in terms of the names of varieties they identified, as well as the frequency at which they identified that. The method E and F, which were expert opinion on either showing them the photos or the seeds, again we got a little more diversity in terms of the names of the varieties, and also more frequency for some of the varieties. So this just shows the variation of responses you got, we got across the different methods that we had tested. Now this is the result of our DNA fingerprinting. Across all the 855 or so samples we had, the DNA results could identify about 5 variety types, and the most common variety group was the Kablumgeti, set 3. The next one was Lukupa, Lwangeni and Lambai, and these are the 4 out of those 11 released varieties that were included in the accessions. And what that means is that other varieties, the other 7 varieties that were included in the accessions, there was no match in the farmer samples. Only this 4 varieties there was a match. In terms of the land races, also there was some match with this Kablumgeti variety type, and based on this result, the way we classify Kablumgeti set 1, we consider anything that matches that set will be an improved variety. For all these 4 cases, anything that matches the Kablumgeti are local variety, as well as those that don't match anything in our reference library are also in our study considered as local variety. But I just wanted to add a footnote that we consider them as local variety, but it could be that they may be a released variety in another country and they may have spilled over to Zambia. But if that other released variety was not in the reference library, then we cannot identify that as match that with any released variety. So basically, I think it's better to say that they are non-Zambia released accessions rather than local variety, because it's possible that it could be an improved variety but in another setting. This is the table which shows how effective the different methods that we tested in Zambia are against the benchmark of DNA fingerprinting. So this last column here shows the results of how many data points can be classified as improved variety using DNA fingerprinting. So according to this DNA fingerprinting, out of 855, 141 data points could be considered as improved variety, which means 16% of the sample was improved variety. However, in method A1, that same estimate was only 4%. In method A2, which is asking the farmers what type of variety they planted, the estimate is 13%. Method C had the highest estimate of adoption rate, which is 71%. And to recall, this is the method where we showed them the samples of released variety seeds and asked them to match the thing. The expert opinion based methods E and F, the estimation was close to the benchmark, 18% in the case of looking at the photographs and giving the opinion, and 15% in the case of looking at the seeds. So as you can see, there is a huge variation in terms of the estimates of adoption rates across these different methods, and also there is huge difference in terms of the effectiveness. No one method stands out to be effective because they also have large type 1 error. Type 1 error basically means the false negatives, that local variety was incorrectly identified as an IV by a given method. The method C had the highest type 1 error rate. In terms of type 2 error, where an improved variety was incorrectly identified as a local variety, again there is a huge error rate across all the methods. In terms of the accuracy of name and accuracy of category, also no method can be considered as effective if you just look at those low accuracy rates or high inaccuracy rates, I guess. So the main findings from the Zambia case study is that there is no one method that stands out to be the most effective across all measures. When you simply ask farmers or experts for the name and type of bean variety the farmer was using, they tended to be under-reporting that. So there was an underestimation of adoption rate based on that method. When you showed them the seed samples, they overestimated the adoption of improved varieties, and this was especially the case because we only had shown samples of improved varieties, and perhaps this is one of the shortcomings of the study, that we should have included maybe a mix of both improved as well as traditional varieties, so they had more options to choose from. So based on those results, one can say that maybe the expert elicitation based methods, where they can look at the photos or the seed samples, gave the most closer estimates on the adoption rate. But again, these methods also had high type 2 errors and low accuracy rates compared to the benchmark of DNA fingerprinting. So for Ghana, a very different story. We had about 180 variety names reported by farmers across the 914 accessions that we collected from them. So you can imagine the huge diversity in names by which farmers were calling their varieties, and some of the popular names that came out are in this figure, where the font size of the name of the variety is proportionate to the frequency at which it was reported in the database. So Ankara, Debor, Banke Coco, Bosomensia, these were some of the most frequently cited varieties by farmers. In terms of the type of variety, farmers categorized their cassava. 51 out of 914 were classified as improved varieties, 796 or almost 800 as local varieties, and 67 as no response or don't knows. Again, very low, if you just go by this, a very low adoption rate. Only 51 out of 914 were grouped as improved varieties. These are some of the names of the varieties that were identified by farmers, and also the frequency at which they were identified in our sample. The most frequently used name by the farmer was agric, and basically agric, I was told, means that it came from an agricultural research institute, and they just short formed that as agric and were calling it as an agric variety. And then there were some other varieties with local names. The ones that are highlighted in yellow are the names of varieties that are in the actual released variety list. So those are the names of cassava varieties released in Ghana. Okay. In terms of the identification of varieties based on field visits by the cassava expert, they identified the varieties by 86 unique names, and the most frequent in their estimate was Debor at 98, Bosomensia 62. So many of these are also the popular varieties that were identified by farmers, but the order or importance is different than what farmers had given. About close to 300 observations, the experts were not able to identify them by names. So even though they are experts, or they were experts, they couldn't identify those cassava varieties by names. Maybe I'll skip through some of this so we can speed up. Again, very interesting, but a very different type of result of DNA fingerprinting for cassava. What this table shows is how the DNA results were used to classify the farmer samples into unique varietal cluster groups. Okay, so there were 11 unique varietal clusters that were disaggregated or differentiated from each other based on the DNA fingerprinting result, and each variety group I've sort of listed the frequency at which the samples fall in that group, and both in terms of farmer sample and the library accession, the reference material. So as you can see, variety group 1 has all land races included, so they are clearly a land race variety group. Variety 2 had both the released varieties as well as some land races included in that group, and about 158 samples from farmer field matched that variety group. Similarly, there are some other variety groups where there are released varieties as well as local land races that matched, and there were several where there was no match, which basically means that they are at least not released varieties in cassava, but they could be local land races from other countries that have come to Ghana, or they could be varieties that are local in Ghana but were not included in the reference library. The DNA fingerprinting also identified several hybrids or admixtures based on their analysis. And basically what these hybrids or admixture groups mean is that they were identified based on the percentage of ancestry belonging to those 11 unique varieties. So there was, if the accessions matched, had 50% of ancestry coming from variety 1, they were uniquely identified as one of the admixture group 1. If it had 50% ancestry coming from variety 2, it was identified as admixture type 2, and so on, till the other admixtures which were more of different variety mixes together. Again here, the some group included just the improved varieties, and some included just the land race materials. Okay. So this is a very complicated result table, but the main, the bottom line of that table that I want to point out is that there were three interesting findings based on that DNA fingerprinting for cassava. One is that, as I pointed out, there were multiple improved varieties included in the same varietal group, which meant that more than one improved varieties were genetically identical. Okay. The second interesting finding was that many released varieties were hybrids or admixtures, as I showed in the previous page. And third, which was the most critical from our perspective, was that library accessions representing both released varieties and land races were part of the same varietal group or the cluster group, and there were four such groups, that variety group 2, 3, 4, and 8. And this particular finding poses the most challenge for our work in terms of varietal identification. This is because it raises the question of how do we classify the farmer collected samples that fall in those categories, in those variety groups. We consider them as improved varieties, or do we consider them as land race varieties? Yeah, so that's a challenge that we faced in interpreting this data and applying it for our study. To address this dilemma, the way we have tried to tackle, or I have tried to tackle this, is to look at two scenarios: a liberal assumption scenario and a conservative scenario. In the liberal scenario, we sort of classified all the samples that fell in those categories where there was at least one released variety. We considered that farmer sample as improved variety. In the conservative scenario, we considered that group as a traditional variety. Okay, so you can imagine these are two sort of extreme end of the spectrum of whatever the truth was, and we present our results sort of based on both these scenarios of how effective other methods were. So let me quickly just go through this slide, which shows the liberal assumption scenario. According to this assumption, about 284 data points can be classified as improved varieties, which comes to about 31% adoption. Yeah. Compared to this 31%, other methods gave 1%, 6%, 2%, 5%, and 15%. So you can see a big gap in the estimate of classifying improved variety adoption rate using DNA fingerprinting versus these other methods. Again, a very high rate of type 1 and type 2 errors across the methods, so no one method sticks out to be the most effective. In the conservative assumption scenario, the number of data points that can be classified as improved variety falls to just 40 observations. And what this means is that only 4% of the samples can be considered as improved variety. And now that 4% seems more comparable to the estimates we get in other methods, the 1%, 6%, 2%, and 5% under methods A1 to D2, and the 15% method under method E. Okay. So the main findings from the cassava case study is that no method sticks out to be or stands out to be the most effective on all measures. On aggregate level, adoption of IV methods based on farmer elicitation and field observations by an expert provide the closest estimate under the conservative scenario, but again it has very high error rate. Thus, no method comes closer to the truth in adoption estimates under the liberal scenario. Identifying cassava varieties accurately by name is most challenging in a setting where there are hundreds of variety names that exist in the farming system. And again, adoption estimates by experts based on photographs gave a substantially higher estimate than the other methods, but this method had a higher type 1 error rate. So based on these studies, what are our key methodological implications for tracking varietal adoption? First of all, relying on for both what the beans and cassava case study has shown is that when there is a diversity of names in the farming system and farmers call the varieties through diverse names or different names, the traditional method will give you an underestimate of your adoption of improved variety, yeah, by name, if you are looking for name. And thus what we have so far considered as the gold standard of eliciting varietal adoption based on farmer surveys may not be the best suited method or give you an accurate estimate when your interest is in measuring varietal turnover or in assessing the, the benefits you get from plant breeding research from farmers changing newer varieties and replacing it, replacing newer varieties to the older varieties. When your interest is in measuring variety specific adoption, maybe showing seed samples is a good method, but it can lead to an overestimation of adoption if you are not careful in what types of samples you have included in the display that was used. And this was one of the limitation of our study, and perhaps more studies are needed to correct this and to test what that upward bias could be in this method and how that can be reduced. The methods that measure adoption of improved varieties, the farmers and experts are better able to give an aggregate picture of what the adoption of improved varieties is rather than variety specific adoption. However, these methods are prone to both type 1 and type 2 errors. So, it has implications on how the accuracy of any analysis you do on data that uses this kind of method. Let me just quickly go through some of the conclusions regarding DNA fingerprinting as a method. What our study has shown is that this could be a useful tool for determining the genetic identity of varieties grown by farmers, but it can give a true picture of what is out there in the fields. However, as the case of cassava has shown, the truth can sometimes be ambiguous, especially when in the setting that we had, where both the improved varieties and the lane race varieties were genetically identical. Then you face this dilemma of how do you classify the farmer collected samples for such groups. Okay. There are also potential scaling up challenges in using DNA fingerprinting for varietal identifications, and some of these challenges relate to the logistics of doing this study in farmers' fields, the cost of doing DNA fingerprinting, and also the need for capacity to do the DNA extraction, the analysis, the interpretation of data and analyzing it and things like that. So given these challenges, at least personally I think that the use of DNA fingerprinting is part of a representative survey is far from becoming routine right now. We need to find more ways to reduce, first of all, the cost of doing DNA fingerprinting and also making the logistics more manageable so that in future this can become a more routine thing that can be done as part of the adoption surveys. Also, more studies on different crops and country settings are needed to generate more experience base and to derive some generalizable results. So I fully agree that whatever I have made some conclusions or recommendations here are just based on 2 pilot studies, and more studies are needed to validate some of these lessons. Thank you very much. Thanks for your time.
Thank you. Now floor is open for questions.
Thank you very much for a very elaborate presentation, a nice presentation. Regarding your conclusion 1 and 2, the large part of the result depends on the nature of the crop. because the beans you selected, the cassava, they are phenotypically difficult to distinguish even by expert. So whether farmers can or they identify or not depends on the nature of the crop. Because this, because they are so the conclusion of that, it also for that observation depends on the crop. What is crop you are selected? Like a beans, if you see all the beans are looks like, say, even expert cannot distinguish which variety, by seeing the plant or these things.
No, in beans we didn't use the plants. In beans we only use the seed samples.
Yeah, that's why because seed you higher there, because seed is only different variety, different only seed only can be distinguished.
Yeah. But in case of cassava we use the plants.
Yeah. So this is that's why the what is called the conclusion are very crop specific. That's why
Sure.
crop determine your, because though they are generic conclusion, but generic conclusion cannot be done by the two crop only because the crop influence is high. That's one point. The second point is how this can use for the general behavior or any economic part of a study. What
can be used for
generic or economic behavioral conclusion, what we can draw. Because are we saying by that adoption study is not good by journalists, because there are so many, this is so much study done on adoption. It is one of the largest number of socio-economic study done.
That's right.
So what is the conclusion on that?
Sure. So basically what our studies have shown is that in the past we have relied on asking farmers what varieties they have adopted, and all our socio-economic analysis is based on that response of the farmer. Our adoption rates are based on that response. Our economic analysis on determinants of adoption, what factors determine the adoption of varieties by farmers is all based on that response by the farmer. To me, this study, I know this is only for 2 crops, and more crops are being done by other researchers, and we will get more information on that. To me, that analysis, it raises question on the validity of those analyses if there is a systematic bias in farmers reporting knowledge of the variety and their socioeconomic status. So the further steps we are going to do with this data is to do the similar type of analysis based on the results if we were to only use farmer response. What are the determinants of adoption, for example. What is that? We already shown that the adoption rate is very different if you use farmer response versus DNA fingerprinting. So we are planning to do more analysis showing the implication of this finding in terms of the socioeconomic conclusions that we derive. Yeah. So that's still work to be done on that. Yeah.
It's like more like economic study only. What is the economic policy we can draw?
Sure. Which has still to be done. Yeah. Agree. Yeah.
You have results for DNA fingerprinting as well as from socio-economic, like household surveys, right?
Yes.
So did you come across, like, for a particular improved variety from household survey you got some figures, say 84% this improved cultivar is grown in this area. When you did DNA fingerprinting for those samples and you got different some type results, means, and like how much it was mismatch?
So
how much, like
so in the case of beans, if I may go back, this is what the mismatch is. Anything you see here, so this is, if you consider this as the truth, 16%, whereas anything, all these numbers, if they are different from that truth, that is the discrepancy you are talking about. So if we were to just use the farmer survey and do any estimate of adoption, it will be only 4% of the samples would be classified as improved variety versus 16%. So it will be an underestimation of the adoption rate of improved varieties.
So do you think that maybe if it this rate of you can say DNA fingerprinting based survey. If it is little based higher also, it provides more you can say reliable results. So
What was higher, sorry?
you mean to say, suppose would you like to summarize that DNA fingerprinting based surveys, although they will be little bit higher
In cost?
cost.
Or, yeah.
But they provide the more relevant and precise results.
I mean that was the whole purpose of using DNA fingerprinting as the benchmark, because we know that gives you the most reliable and accurate result. The only thing is it's costly.
Yeah.
And so we wanted to test these other methods to see if there are other cost-effective ways of getting the same results. But what our study has shown is that there is no one method that stands out to be the most effective. And thus, as of now, if we were just to look at these results, it seems like we should include DNA fingerprinting in all our farmer surveys. That's what I would conclude. If budget was not a constraint, I would always use that as a method.
So maybe you might have taken account the cost comparison in different methods?
No, not actually the cost, but we know all the logistics that goes into implementing some of this method, and we have also estimated the time it takes for the enumerator to visit the field, collect the sample, for them to do the photographs, to show them the samples. So all that, there is a cost element, there is a logistics element, there is a enumerator training and time and all that implication. That's right. Yeah.
This is what I feel. So if you will have this kind of cost comparison, then you will end up in the DNA fingerprinting.
That's right. No, you're very correct. You're very correct. That maybe DNA fingerprinting, also has some logistics and cash cost, but compared to others, maybe it's not that bad. Yeah.
The relative proportion between type 1 and type 2 error varies between beans and cassava. So, what do you think is more dangerous? I mean, normally we say type 1 error is more dangerous.
That's right. That's
and
right.
then has it to do something with the method by which these crops are being propagated? One is sexually propagated and the other one is,
It's
the
just
way in
a
which it is
seed
grown
based, yeah.
and the crop duration also. So how does that figure in the combination of type 1 and 2 error?
Yeah.
What is the conclusion from that?
Yeah, so to me, that the crop which has more diversity, as it was in cassava, we got 180 unique names of varieties that were planted. You are prone to have more error, because the probability of the name actually matching is very low. So I in a setting such as cassava, where there is a lot of local names or variety names prevalent in the system, it will be very difficult to just rely on asking farmers or farmer elicitation based method of varietal identification. And I was planning to also do a study on maize, where maybe because of hybrid and more matured seed system, farmers may have more better knowledge of varieties they are planting, because if they are more used to buying seeds, then they would know the names of the seeds, varieties, more accurately, and there will be less type 1 errors in those situations. But of course, both cassava and beans are in a setting where the seed system is not matured and farmers have not been used to buying seeds either for beans or cassava that much.
In fact, my experience is that adoption study is getting more traction where more number of smallholders are there, and more number of players in seed system is also there. So there is a lot of confusion and mess, and everyone wants to know that how much impact we are creating, one research organization, one research group and another research group, right? So there is always fight that NAR system is saying that our variety is more, and other system, organization is saying more, our varieties are more in the field. What happens that where, as you have said, that in cassava more confusion is there and mismatch is a little more, because land races are more there.
Yes.
Where you can't find that established players in seed ecosystem are there, right? But in when we have seen in maize in India, what we have observed that wherever hybrids are there, farmers are a little more knowledgeable.
Yes.
And there is no need of having DNA fingerprinting. It is very much obvious
That's right.
that which company is ruling in that area and in hybrid.
Yeah.
So wherever land races are there, means those are underdeveloped region, and farmers are also very poor and they cannot afford those means costly variety. So my issue again comes to the cost, that how much feasible it is there to upscale this kind of technology to have. Definitely DNA fingerprinting will give more accurate result, but is
But
it
it's scalable.
more overriding on the farmer's experience? Right.
Yeah. So our pilot study, the cost estimate for DNA fingerprinting came to about $30 per data point per sample, not sample, but yeah, per sample I would say. Yeah, per data point. So like for 900 data points, per data point $30 was our estimate. And this includes the cost of extraction, analysis, I mean the sequencing as well as the analysis component. Yeah, researchers' time cost, well, we outsourced the sequencing part. So, you know, we just received an invoice. It was a service, cost of service. But the analysis part, yeah, it's a researcher's time which may be subsidized by somebody because it may not capture the full cost of the scientist's time. Yeah.
I think cost may be per sample is $30. Data point, because it gives GBS gives.
No. So yeah, let me point. Yeah, it's not sample times snake.
Yeah, exactly.
So it's per. So when I use the word data point, I mean this 855. That's what I mean. Observations. Yeah. No, there was no specific purpose except that we wanted diverse crops, and then there was an opportunity in Ghana. Somebody was doing some study on cassava, so SPIA, who is funding this study, told me that why don't I do this study in cassava in Ghana. So I ended up doing that, and then I learned about bean study being done in Zambia, so I selected that as the crop. But we have also selected maize in Uganda, and we had budget to do about 2 or 3 studies. So
luckily beans has different kind of seed pattern also, but in other cereals you may not find.
Yeah, that's right. I mean, beans has different market classes and shape, size, color, everything. Yeah, that's right. Yeah. Sure. Yeah. So I plan to do, and that was one of the purpose of my visit to ICRISAT, is to do something in India on wheat and lentils using DNA fingerprinting. So I'm sure there are different crops than cassava and beans.
So in this we come to the conclusion for today.
Okay, we can continue.
So I think we can have a round of applause and thank you, Mywish.
Thank you.